Inflammatory bowel disease involves chronic relapsing intestinal inflammation. IBD is thought to involve complex interactions between genetic, immune-based, microbial, and environmental factors. In IBD, dysfunction of both innate and adaptive immunity contributes to the intestinal inflammatory response. IBD is characterized by enhanced recruitment and retention of effector macrophages, neutrophils, and T cells into the inflamed intestine, where they become activated and subsequently release pro-inflammatory cytokines. The JAK-STAT signaling pathway is a universally expressed intracellular signal transduction pathway that is involved in many critical biological processes. Increasing evidence indicates that the persistent activation of JAK-STAT signaling is closely related to many immune and inflammatory diseases. Activation of this pathway begins with cytokine binding outside the cell. JAK proteins associated with the cytokine receptor on the interior of the cell then phosphorylate each other. The phosphorylated JAKs, in turn, phosphorylate tyrosine kinases on the receptors, creating docking stations for STAT molecules. After STATs dock, the JAK molecules phosphorylate the STAT molecules and they dissociate from the receptor. The STAT complex undergoes dimerization and translocation to the nucleus, where it binds to DNA and initiates DNA transcription. This results in cell proliferation and differentiation, and the inflammatory response in IBD. While biologics that block various upstream cytokines have emerged over the past decade, agents that inhibit JAK proteins downstream have also become attractive targets for the management of IBD and other inflammatory conditions.